I would like to thank everybody for being here, and I'd really like to thank the organizers. I realized last night when I was putting this talk together that probably if I'd thought about it a month ago, I might have used a slightly different title and a slightly different um, abstract. But what I really want to do today is honor Carl's memory by um, giving you a flavor of how important theory has been in guiding our experimental work. And I think that's the best way I can think of to honoring a very, very wonderful and talented theorist, is to show you how important theory has been in our experimental work. And I want to just spend 1 minute talking a little bit about Carl. I was very, very fond of him. He was a graduate student in Larry Abbott's lab, and we had at that point in the Volin Center, we basically shared lab space. And so I used to see Carl most days. And he was quite an unusual young man. I think it's likely that I taught him his first neuroscience course. And I only realized much later in thinking about it that he probably couldn't see a textbook, but he could listen to me. And I think he probably remembered everything I said, but when it came time to take exams, he only wrote down the answers to the problems that had quantitative answers and didn't bother writing down answers to questions that needed a lot of words. Carl used to, in those days, he was already pretty visually impaired, but this is way before we had the politics of making accommodations. So I think he just went about his life and he did his math in his head, and he squinted at the screen, and he never ever complained. And he was just quite a wonderful person. I'd just like to give you one more anecdote about Carl. So several years later, we had a picnic on the banks of the Charles, and it was joint picnic with my lab and Larry's lab, and we were sitting there, and all of a sudden Carl rolls up on roller skates, and he was careening from side to side with arms and legs flailing. And we said, Carl, we had no idea how he had gotten the 5 or 6 miles on rollerblades from where he lived to where we were. And he said, yeah, it was his first day of ever using the rollerblades, and so he was still learning how to skate. And I just remember being so terrified that he was going to be hit by a car. I said, do you want us to give you a ride home? He said, oh, no, I'm just going to go. And I think that joyful fearlessness, despite his visual impairment, is one of the things that I really cherish so much about knowing him. He was quite a gentle, lovely person, and I was very sad when I heard that he had died. Anyway, so as I said, in honor of Carl, I'm going to focus on some illustrations of how important theory has been in guiding our experimental directions. And I could go way, way back, but I'm not going to go all the way back. But I'd just like to, before I forget, to show you this is my present lab up there on the left, and all the wonderful people with whom we've worked. And I just like to say that regardless of where they've ended up and what they're doing, I'm incredibly proud and grateful to have had the chance to work with so many wonderful, smart people. And this is my present lab, and I can almost see Carl in there making crab signs with his hands, because my lab has always been full of quite quirky people, and Carl certainly was a quirky person. Anyway, to get going, what we've been working on in the last 20 years is basically what I call trying to understand the role of degenerate mechanisms or multiple mechanisms that give rise to very similar behavior. And we've been studying this using both computational and experimental work. And we came to these understandings basically first driven by computational work, and then that drove us to do some experimental work. And I'm not going to go all the way back to the beginning, because the beginning really was embedded in our first homeostatic models. But I'm going to start first by showing those of you who don't know it, the preparation we use. And this is the crustacean stomatogastric nervous system. And again, for those of you who don't know, this preparation was developed in the 1960s when people interested in trying to understand how circuits worked were looking for preparations that would produce in vitro motor patterns in this case that resembled very closely the motor patterns recorded in vivo. So just to show you that, this is a crab. The crabs we work on are about so big. They're 6 or 7 inches. Okay, certain things I still think about is in inches, not centimeters, but inches big, and they're 5 to 7 years old. And this will become very important later for you to realize that these animals have survived in the North Atlantic Ocean, just outside where I live, for multiple years. And so what you can do to record in the intact nervous system is make a hole in the carapace and put a suction electrode on one of the motor nerves. And if you do that, and then you can seal it back up and put the animal back in the tank and record now in vivo these motor patterns. And this is what we call the pyloric rhythm. It's a triphasic motor pattern. And this rhythm persists throughout the lifetime of the animal. And therefore it's very, very attractive. And because it's very stereotyped, it's also very convenient because it's very easy to see when it's working correctly and when it's been disrupted or damaged. So in this motor pattern, this triphasic motor pattern, you first see a discharge of a neuron we call LP, and then a neuron or a set of neurons we call PY, and then a set of neurons we call PD. So the motor pattern is LP, PY, PD, LP, PY, PD, and in the animal, it has a canonical frequency of about 1 hertz. And now if you then, and this you can see it's very, very regular, and that's actually very convenient. And could I ask, Sarah, could you turn off your camera because you're moving around and it's distracting me? Thank you. So you can next do is you can remove the stomach from the animal and dissect the nervous system off the surface of the stomach and now record in vitro. And when you do that, what you see is a motor pattern which is very, very similar to what you see above. Now this is the in vitro motor pattern, and this is an example now of a central pattern generator that can generate this rhythmic motor pattern in the complete absence of rhythmic sensory input because it's gone. So there's no sense that this is a fictive motor pattern. There's no sensory inputs left, although there are some descending modulatory inputs. Now, the neurons that are most important for producing the rhythm are 3 neurons. The anterior burster AB is an interneuron, and then there are 2 PD neurons, and they are electrically coupled to the AB neuron. So this is the pacemaker kernel, and the PD and the AB neurons together rhythmically inhibit the LP and the PY neurons. And the important thing here is that this rhythm exists in the complete, or this rhythm occurs basically on the basis of inhibition as the follower cells fire and rebound from inhibition. So unlike many systems which depend heavily on excitatory synapses, this is really driven by electrical coupling and inhibitory synapses. So this is the preparation that's going to be featuring in some of the experimental and computational work that I'm going to show you. Now, somewhere, why is this no longer working? That's not good. What have I done? What have I done to destroy this? I will no longer advance. Okay. Yeah, okay. So in the models I'm going to be showing, all the models I'm going to be showing you have very similar flavor. Now, you all know that neurons have many different kinds of voltage and time dependent ion channels, and I in my lab have spent a lot of time characterizing those ion channels. As all biophysicists who try and understand ion channel function use voltage clamp measurements to look at the opening and closing of ion channels, and then to measure the maximal conductances and the the rates by which the ion channels open and close. Now, and those data then fit with differential equations, and then they're turned into a model that just expresses the relative contribution of those voltage spin currents to the dynamics. Now, the models I'm going to be using have an inward sodium current, 2 inward calcium currents, an IH or hyperpolarization activated inward current, 3 potassium currents, a delayed rectifier, calcium activated potassium current, and a transient outward potassium current, and then a leak current. Now, the models are isopotential, and that just makes it easier, but it turns out because of the way our cells are built, these isopotential models do pretty well at capturing the dynamics of the cells. So as I said, you should remember that these are now, in terms of the computational community, for sort of complicated, detailed models. For some part of the community, for other parts of the community, they are definitely oversimplifications. But to me, as a neuroscientist, it's very important to maintain models that treat ion channels correctly, and that models that actually compute in conductance. And so these models are actually sitting there in what I call a sweet spot of being semi-realistic. They're not really realistic, but they capture the essential features that I consider important for neuroscience. Okay, now this story, the particular story I'm going to tell you today, starts with a paper that many of you have read and many of you know about. And I usually don't often show this anymore because it's fairly old. This was a wonderful study that Astrid Prince did. This particular paper was published in 2004. There was an earlier paper that was published in 2003. And Astrid, when she was a postdoc in my lab, wanted to build a good model of the pyloric rhythm. And she wanted it to be, again, semi-realistic. So she started out hand-tuning a model and got very frustrated. So she decided to use what she called brute force. So she started out with a model neuron like the one I showed you before. And she chose, I don't remember whether it was 6 values for each of the 8 conductances in the model. And then she put it all on the grid, and she simulated more than 1 million values of those. And then she asked for each set of parameters what the model's behavior was. And some of them were oscillatory, some were silent. They were all different kinds of cells. And so she selected through that database for models that she considered were good representations of the pacemaker kernel, the LP cell, and the PY cell. And she selected 5 models of the pacemaker, 5 of the LP, and 6 of the PYs. And then she took the known connectivity, what we would now call a connectome, and she chose 5 or 6 values for the synaptic strengths. And then again, she simulated all against all, and she simulated more than 20 million model networks. Now, at the time, remember, this was 2003, this was a pretty large undertaking, but the models ran on the cluster by today, a very slow cluster downstairs. But eventually, Astrid produced all these models. And then she asked which of these models were good models for the pyloric rhythm. And so here was her all networks. 19 of them, 19% were pyloric-like. And by that, Astrid meant that they were triphasic in the right order, LP, PY, PD. But only 2.4% of them were what she called good pyloric models. And what she meant by that is that they had the appropriate frequency and phase relationships that came from, in comparison with experimental data. And these are experimental data that Dirk Bucher had collected. And she used these experimental data as criterion to say, let me find all models that fit within the range seen in experimental data chosen from 99 papers. So this was this was really quite revolutionary at the time, because instead of trying to fit one model that would try and fit the mean of experimental data, Astrid was looking for a population of models that would fit a population for distribution of experimental data. And I think this is why this paper was so useful and so influential, because it really changed the way we thought about creating models. Now, this next slide is one that I'm sure many of you have seen. And when we were doing this, I asked Astrid to find, because she noted that there were many models with very similar behavior. And I asked her to find 2 models with very similar behavior, but with different conductances. And it took her about 3 hours to find these models, which told me that it was a very easy thing to do. You know, when you ask a postdoc to do something and it takes them 3 weeks, it's one thing. If you ask a postdoc to do something and they come back in 30 minutes, you know, it was very easy. Anyway, this was quite easy for Astrid to do. And this then became this very famous picture. And so this was model network 1, model network 2. They're very similar, and yet they have very different underlying conductances. And some of those conductances are shown below. Here's the PY neuron, here's the H current. In the PY, on this one, it's big, on this one, it's little. And likewise, there are all these different sets of conductances. So to me, this was really exciting. And to almost every experimentalist I knew, this was very, very exciting. And it was very exciting because it said that a lot of the variability that we were routinely seeing in our experimental data might really be consistent with reality, because, you know, all the variability we'd always seen before, we all worried about whether we had introduced the variability just as a series of measurement errors. But instead, this said that it was possibly potentially consistent with, as I said, with reality. So when I started talking about this work, as I said, the experimental community was very enthusiastic. The theory community, however, pushed back. And they pushed back in 2 ways. I would get one pushback, which said this can't be true because it's not optimal. There's no single optimal, and everybody knows the biological systems were optimal. And I used to just push back at that and tell them that since when were they optimal, and people would laugh, and that was the end of that. However, I got a very important critique from the theory community, and I really have to thank the next 20 years of the work from my lab for this critique that came from your community. They said, correctly, that although this behavior and this behavior look very, very similar, because they have very different underlying sets of conductances, they will, by definition, respond differently to different perturbations. And therefore, how do I know that this is at all sort of a reasonable way to basically build an animal? So I started thinking very seriously about this, and I realized that what the theory community was asking me to do were 2 things. One was to measure the variability across animals, in individual animals, of the many of the parameters that give rise to this behavior. And 2 was to do a series of global perturbations that would ask whether animals with different sets of conductances or different parameters could respond reliably to those properties. So what we did is both of those things. And I'll put this out of these. So first I'll show you this and say, how variable are the conductances in biological preparations? And these are just recordings that Marder, who was a graduate student a few years ago, made from animal 1, animal 2, animal 3, animal 4. From these intracellular recordings from the LP and the PD neuron, and an extracellular recording showing that triphasic motor pattern. Animal 1, animal 2, animal 3, animal 4, you can see they're very, very, very similar. And Mara didn't look very hard to find these recordings. These were just in her baseline recordings, and almost everybody in the lab who is reasonably competent could have produced a set of recordings that look like this. But when we, and this work started in about 2006 with Dave Schultz and Jean-Marc Gaillard, who were then postdocs in the lab. This is a slightly later study. We take single PD neurons and dissect them out, in some cases after doing biophysics to measure the conductance of channels, or else just directly, and now use single cell PCR to measure the mRNA copy number. And this now is a list of a whole bunch of ion channel genes. What you see is a range of values, and it's usually about 2 to 6 or 2 to 7 fold. And we see this 2 to 6 or 2 to 7 fold range in parameters in an identified cell across animals. And it's what doesn't matter whether we measure mRNA levels or we measure synaptic strength using electrophysiology, or we measure the amount of current in voltage clamp across animals. So the take-home lesson from these experimental studies that we started, as I said, in the first few years after I had that pushback from the theory community, was that individual synaptic and intrinsic parameters can vary 2 to 6-fold across animals, even when their networks show very similar behavior. Now, at the time, the whole experimental community was pooling their data and publishing only means and standard errors. But I started urging people, and other people started urging people to sort of unpack their data. And this 2 to 6-fold range shows up in almost every preparation, in almost every single data set. And I think it's because this 2 to 6-fold is about as good as biology can do, and it's good enough to create these degenerate solutions to maintain constant function across variable systems. So to move to the next step, the question of whether perturbations of various kinds can reveal differences in degenerate neurons and circuits, and when and how do these differences matter? So we started working on a series of global perturbations that were intended to influence all circuit elements. We didn't want to just look at individual elements one by one, but we wanted to influence basically the whole parameter set. And so we spent a lot of time studying temperature, high potassium, pH, and salinity. And these, of course, are the high potassium so much. High potassium is very important in the human nervous system or physiology, but temperature, pH, and salinity are very important to a crab out there in the world in the ocean. And you'll see how this eventually led us to climate change. So what we set out to do was to use this very stereotyped pyloric rhythm to look at the effects of these perturbations. And we reasoned that if, as the theorist said, that any tiny perturbation would be inconsistent with animals having robust or reliable outputs, we would see that. But we wanted to know whether strong perturbations that are perturbations that the animals had evolved to deal with, whether they could respond reliably to those perturbations despite that 2 to 6-fold variability in all of the underlying circuit parameters. So the first thing to study that we studied actually historically is temperature. And temperature, so the other thing I have to tell you, if you don't know, is that every single protein is temperature dependent, and this is an automatic response because the protein itself, the kinetics of the protein itself, will be influenced by ambient temperature. And biologists often use a value called the Q10 to characterize the temperature dependence. And the Q10 is described here. Notice that there's an exponential relationship. And basically, a Q10 of 1 means you're temperature invariant, and that's very, very rare for any biological protein. Many proteins have Q10s of 2. Some have Q10s of 3. Most ion channels have Q10s of 2 to 4 if you look at both the conductance of the channel or the activation and inactivation rate constants. Now, if you think about it, and again, the theorists and you should immediately realize that this would be a problem. If you had a neuron that had an action potential that involved opening and closing of sodium channels and potassium channels in a certain sequence of activity to give a certain voltage against time waveform, it's very, very easy to imagine that if you now go from 10 degrees to 30 degrees, and if each of those properties of the proteins have different Q10s that this now very nonlinear system would become inoperable, or you'd start seeing things mess up. And this is just a slide that illustrates that Tim O'Leary made for a paper that he published on temperature in 2016. So Tim had a model which is very similar to the one I showed you before. And he implemented 3 different sets of Q10s and 3 different versions of the model. At the reference temperature, here you see here, all 3 of these cells were bursting, but the first set of Q10s, as the temperature was raised from 5 to 25 degrees, the cell was bursting slowly, and then it went into a 1-spike burst, and then it went into a depolarization block. This set of Q10s actually was able to increase its frequency and maintain approximately constant behavior across that temperature range. This set of Q10s produced a single spike burster and then went bursting and then made spikes and then blocked in a hyperpolarized. So, and it turns out it's not easy to find sets of Q10s that will maintain consistent behavior across a range of temperatures. So that means evolution has had to work very hard to do this if it can, and I'm going to show you now that it does do this. So these are data. This was the beginning of our temperature work, and it goes back to work that Lamont Tang, who was a graduate student in the lab, did, and he took a pyloric rhythm. These are extracellular recordings where you're seeing PDLPPY, that's the triphasic rhythm that you've grown to love. And this was at 7 degrees, and this was at 19 degrees. You can see the rhythm has gone faster, which is what you'd expect. But the really important thing is that the triphasic rhythm remains completely intact. And so this tells you that that the crab, which lives, the crabs that we work on, see temperatures as low as 2 degrees centigrade and as high as 25 degrees centigrade, that they have mechanisms that allow them to maintain the triphasic rhythm, even though all the Q10s that are controlling synaptic and voltage-dependent currents are different. And so the remarkable thing, when Lamont brought me this slide, my mouth dropped open, because these are now the phase relationships, that is to say, the relative time that each neuron is on and then off. The phase relationships are completely constant, despite the frequency change that comes from temperature. So now if you think about this, as all of the rate constants are changing, but nonetheless the pattern is maintained, you can see the miracle that biology has found, because it turns out to be a not trivial computational problem. Okay, so this is basically the same story. Now, Lamont is here showing you intracellular recordings with PDLPPY at 7, 11, 15, 19, 23. And you can see that as the frequency increases, as temperature increases, as you'd expect, nonetheless, the phase relationships are maintained. And that's seen here as Lamont did overlays of 7, 11, and 19 degrees. He normalized time away. You can see how the waveforms are maintained. So this was really quite remarkable to us. And but we're going to leave that now, because explaining how this happens takes 20 minutes. But we do basically understand it more or less. But the other thing Lamont found is that if he went to higher temperatures beyond in those days, 23 to 24 degrees, he got what he called crashes. And these are crashes in 4 different animals. And notice what he called a crash was when he lost the normal triphasic rhythm and he got disordered rhythms. And the reason I'm showing you this is these are 4 different animals and they're producing 4 different disordered rhythms. And this is exactly what you'd predict if you had 4 animals and they had different underlying sets of synaptic strains and underlying different maximal conductances for the ion channels. You would predict that when it became disrupted, that it might become disrupted in different ways. I mean, this would immediately come out of the fact that those degenerate solutions that were healthy and normal were nonetheless different. And this is what the theorists would have predicted and did predict. And it turns out it only happens when you go to extreme perturbations. And this is just another way of showing you this. This is work that Leandro Alonso did, where he used genetic algorithms to find pyloric rhythms that were capable of producing constant values, constant pyloric rhythms at 10 degrees, 15 degrees, 20 degrees, 25 degrees. But he then asked them to basically crash at 30 degrees. So this tells you, and this is just the way he ran the algorithms. And so really nice rhythms up to 25 degrees and crash at 30. And then Leandro just made this slide just to compare the kinds of crashes that he was seeing at high temperatures in these different models, and just to show you that they are different from each other in the same way, as each of these models has a different set of underlying parameters, and they're crashing differentially with all sorts of weird slow dynamics in a way very similar to what the experimental data show. Okay, so our theorists 20 years ago were right, sort of. Now I'd just like to show you another one of the results that came out of Leandro's work. And what he did, and one of the other things that he did, is he came up with a really nice way of displaying what each of the currents in a model is doing. And I skipped the slides that describe this, but I just will show you that in this way of displaying, all of the outward currents are shown here, all the inward currents are shown here. This is the total outward current. And now we can look in color at the relative proportion that each outward current is contributing to spike repolarization. So here's spike repolarization of a model neuron at 10 degrees. And now there are Q10s on all of these. And here you have it at 25 degrees. It's one of the models that Leandro created using his genetic algorithm. And what I want you to see here is in purple, you see the A current. And now as we go up to 25 degrees, you see the A current. And in orange, you see the calcium activated potassium current. And you see it's big here, but it was quite small here. So I actually love this slide. It was one of my favorite slides of that year. And it's one of my favorite slides because it shows you something that is truly, truly important, which is that these conductance-based models are capable of making smooth transitions in mechanism without losing their activity. So the cell can repolarize its spike perfectly fine, but by 25 degrees, it's repolarizing the spike by a very different mechanism than it was using at 10 degrees. And I think it's very important that the reason why biological neurons in your brain have many different kinds of potassium channels, many different kinds of calcium channels, they're overlapped in time, in voltage dependence and in time dependence, and allows them to make these smooth trade-offs in function so you can slide through mechanism. And I think a lot of the stability in the brain occurs because of reasons like this. And so that's why I'm showing it to you now. Okay. Now, just a couple of other little things from Leandro's paper about temperature. This was published in 2020 in eLife. And these are things that you can see clearly in a theory study that you could never see that clearly in a biological study, but has deep meaning for a biologist. So this was one of Leandro's models with built-in temperature sensitivity. Here's a bursting neuron at 10 degrees, and now 25 degrees. It's bursting faster. And now he can display what's going on with each of the currents. So he's now plotting the sodium current here at 10 degrees, and now here 25 degrees. And you can see it's gotten much bigger. And you can see as we go along, here are the calcium currents and etc. And in red, he's got the currents that got bigger when you go from 10 degrees to 25, and in green, the ones that get smaller. Now, this is another model. This is another bursting model that he created and has different sets of underlying conductances. But now we look at 10 degrees and 25 degrees, whoops. And now you see that the same current is actually decreasing, whereas this one it increased and here it's decreasing. And the position of the green and the red symbols is completely different between these two models. Now, these two models are not that dissimilar in terms of their waveforms, and you could easily imagine two neurons in the brain of the same class with very similar voltage sort of recordings, and yet the model allows you to see very clearly that the effects of temperature could be completely opposed as we look not at the voltage waveform, but at the contribution of individual currents. And the thing is, if experimental neuroscientists don't know and understand this, it can be very confusing as you try and understand the impact of any sort of perturbation or pharmacological agent. And this is just another example of that take-home message. This is a model, again, from Leandro's work, where the A current was at 100%, and at 10 degrees, he blocked the A current, he turned off that conductance, and you can see it's completely disordered. But when he did the same experiment at 25 degrees, you can see that basically the network, here, this is the full network, the network was more or less unchanged. And the opposite thing is shown here. This is the calcium activated potassium current. In this model, the network is perfectly fine at 10 degrees when you turn off that current, but when you do the same thing at 25 degrees, it completely crashes. And this is a really, really important thing for anybody who wants to understand the effects of genetics or genetic manipulations or pharmacological manipulations on real biological systems that are likely to differ in their underlying conductances, just as these models do. So I'd just like to summarize that and say that individuals with similar starting circuit performance with relatively simple circuit mechanisms in the control state can nonetheless show entirely different crash mechanisms. And this really is a very important cautionary tale for thinking about disease, and is particularly important during today's extreme heat events. And I'm going to say that for those of you who are theorists, it can, and if you want to try and explain the diversity or the individual variability in populations, you basically have to make sure that your starting models have elements that have that variability in parameter sets built in. Now, I think I probably don't have time to do this, although it's very beautiful. And I probably will just spend 1 minute talking about this. Now, I said we've studied a number of different perturbations. You're probably curious to what extent do different perturbations interact, because in the ocean, pH and oxygen levels and temperature are all interacting. That is to say, the amount of oxygen that can be stored in water changes as a function of temperature and the depth of the preparation. pH changes, etc., as a function of temperature, etc. So these are experiments that Jacob Ratliff did. He's now recording from the isolated pacemaker kernel, and now in temperature, in experiment 1, he's looking at a bursting neuron, and then he increases the temperature from 26.6 to now to 31 degrees, and you can see he loses the oscillation, he gets a crash, but he has these slow waves all the way down to crash. This is another animal. It crashes at a little lower temperature, as you expect, this diversity of those behaviors, but nonetheless, it crashes by basically the same route. And here's another one, yeah. However, now when he looks at changes in pH, he starts at pH 7 and then goes down to pH. And notice the cells go from bursting to tonic firing, and then they crash. And this happens the same way. So the route from bursting to crash with temperature, high temperature, or with low pH is a very different route. And then Jacob actually followed this up, and this is actually a very pretty paper, and says that here he has multiple preparations. What does that mean? Did I go mute at some point? How far back? I'm okay?
5 seconds ago.
Okay. So we go from bursting to silent at pH 2 as a function of temperature. But when you do a pH ramp at 11 degrees, you go from bursting to tonic to silent. And then this is just further ways of showing that. So I think for the theorists in you, it's very interesting, and this paper actually has a bit of theory in it, to show that the dynamical systems of the transition from bursting to silence goes by a very different path with these different perturbations. Okay, I'm getting close. Why is this not going? Okay, screwing up again. Okay, I'm gonna have to skip these. It's very beautiful. Okay, I'm just gonna end with a couple of slides to the thing that's really, really, really worrying me now. Now, I showed you at the beginning of this Lamont's data showing you that he was starting to see crashes around 25 degrees. When Sarah Haddad started in the lab a number of years later, I told her to repeat the same experiments. And she came to me and she said, they're not crashing. I said, what do you mean they're not crashing? Of course they're crashing. She said, no. So she eventually, after arguing with me, brought me the data. And here you see Sarah's, this was the winter of 2012. This was in March of 2012. 11 degrees, 20 degrees, 25 degrees, 30 degrees, 36 degrees. Now you see the frequency is getting larger, the phase relationships are being maintained, the rhythms are perfectly beautiful, but this is a 6 hertz rhythm. I had never seen a 6 hertz rhythm before because I'd never, usually the rhythm would break down before getting in place. And then we later realized that this was the spring after winter that never got cold. So these animals had never seen anything. And the fishermen down in Woods Hole told us that the squid had all gone to Maine, very sensibly. The dogfish had all gone to Maine, but the crabs had actually managed to adapt to these or acclimate to these very warm temperatures. So a couple of years later, I mean, Jacob Ratliff was in the lab after that. The crash temperatures went back down when the water got colder again. But this was 2021. Marder was in the lab. These data 11, 15, 19, 23, 27, 31, and the 35. And again, the crash temperature was way, way, way elevated. And you can see here, Mara's data. So then Mara did something really quite remarkable. She went back to our lab notebooks, and we do have good records, and she pulled out the raw data from Lamont's data from 2006. Here's 11 degrees, 21 degrees, 31 degrees, just as Lamont had reported. These were Sarah's data, 2012, 11, 21, 31, 35. And these were Jacob's data, 11, 21, 31. Notice this crashed again. And these are Mara's data from 2021, 11, 21, 31, 39. And then what Mara did is she went to the NOAA website. That's the government agency that monitors water temperature. And she looked at the buoy just outside of Boston, and she plotted the average summer temperature in the water in all of these years and the average winter temperature. And first of all, you see the temperatures going up, but that's not a surprise. But this purple dot here is Mara's data, and this green dot here is Lamont's data, and that's true down here and down here. And you can see that the crash temperature, the average crash temperature was quite different in 2016 than in 2020-21, as was everything else, although the pyloric frequency as a function of temperature was pretty consistent across that. So this was telling us that the temperature experience of the animals had produced quite long-lasting changes in their response. But that's not why I chose the title for this talk. This is what I chose. If you just look at 11 degrees, all the networks are fine. This is our usual starting temperature for all of our experiments. And the thing that makes me deeply, deeply concerned and interested is if we had just looked at 11 degrees and done whatever we were going to do, we would have no way of knowing that there had been changes in the network that were deeply profound that would only show up when we did the perturbation. So these are changes I call cryptic. We see very strong cryptic changes when we look at the effects of high potassium. So these now are changes that completely alter the way in which animals respond to subsequent perturbations, but are not evident under normal working conditions. That is to say, not evident until you do a perturbation which will reveal it. And as I said, this temperature experience shows up as alteration in the response to high temperature or the response to high potassium in a really interesting way. So this temperature experience is coloring. It's probably moving all of these networks from one place in that multidimensional manifold to a different place so that they respond to subsequent perturbations differently. And I imagine that this is a way of thinking about people with things like post-traumatic stress disorder, people who appear to be perfectly fine until they run into an extreme perturbation, which reveals that their brain chemistry and their physiology has been altered. And so this is the challenge that we now face. I've become involved with a number of neurologists recently who are studying the effects of high temperature on human neurological disease. And it turns out, and you won't be surprised by this, that epilepsy, seizure disorders of all kinds, stroke, and a whole variety of other neurological problems are highly exacerbated by high temperature. And if you think about it, if you have a network which is functioning okay, but has these cryptic changes in it, and now you produce a perturbing event such as high temperature, you're probably much closer to the bifurcations that take you from okay to not okay if you start out with a disorder network. And therefore, it's not a surprise that people with compromised nervous systems who can function more or less okay most of the time are very, very, very debilitated in high temperature and climate change. And so I think this is what we're facing. All human neurological disease is exacerbated by temperature. And I'm going to just stop by saying that degenerate circuits can hide cryptic differences due to differential history that can alter or shape future responses to additional challenges. And so this is the challenge that we have both as experimentalists and theorists, is to really try and understand which of the degenerate solutions to circuit behavior are fine normally and which are more resilient to the kinds of perturbations that we are all facing. And I'll stop there.